meeting starts. So just give it a moment for everybody in the lobby to actually be into the meeting. Good afternoon, everybody. This is Dominic Zerlo with the Department of Health, and I am just here, and our team from Department of Health is here to administratively help with this committee. We have some people who are still coming into the meeting from the lobby, so we'll give them just a minute to join in. And it looks like everybody is in. So at this point, I'll turn it on over to you, Ian. And Ian Dunn is our chair. So Ian, take it away.
Hi, well, not good morning anymore, but good afternoon, everyone. I hope everyone is doing well. This is the Dosage Administration and Clinical Practice Committee. Thank you all for taking the time to be here. I really do appreciate it. Hey, Dominic, if you are willing to pull up the presentation for me.
Yes, I am doing that right now. Should be on in just a moment. You should be able to see it now. Well,
yes.
Okay.
All
Perfect.
right. Again, welcome. This is the Dosage Administration and Clinical Practice Committee. Today's objectives are to review and adopt our definitions of our dosage levels and just dosage definitions in general that will be relevant to this committee. If you can go ahead and go to the next slide. So just as a quick reminder, in case anyone has forgotten or anyone is new, committees operate in an advisory capacity to the Medical Psilocybin Advisory Board. Committees support the board by expanding community input, incorporating subject matter expertise, and increasing participation in program development. All right, if you want to go to the next slide. Perfect. So again, we will do introductions. So if you wish to serve and participate on the committee today, please share your name, role, or area of expertise and perspective or interest related to dosage administration and clinical practice. I'll go ahead and turn it over to Jorge to unmute.
So if you would like to speak and introduce yourself, please raise your hand. Right. Scott, your mic has been enabled. Please speak when you are ready.
Hello, everybody. My name is Scott Folkman. I am a professor of chemistry at New Mexico State University. My area of expertise is in analytical chemistry and analysis in terms of potency and impurities.
Thank you, Scott. Please forgive me if I mispronounce your name. You can correct me. Laura, your mic has been enabled. Please unmute yourself when you're ready to speak.
I got it. Hi, my name is Laura Martucci, and I am a New Mexico State certified peer support worker. I 100% believe in this program, and I just want to be involved with everything that's going on with it. I have just started a LLC, Fighting for Serenity, and a nonprofit called the Monarchs. And I would love to have this program in my programs. So I'm right here just to listen. So here I am.
Thank you very much. Kate, your mic has been enabled. Unmute yourself when you're ready to speak.
Hi, my name is Chris Piskuski. I'm ambassador for the Heart, and I'm on the advisory board, and I'm just here to help out today. Thanks.
Thank you, Chris. Brenda, sorry, give me 1 second. Brenda, go ahead. Your mic is enabled. Please introduce yourself when you're ready.
Hello, everyone. My name is Brenda Burgard, and I'm on the advisory board for the Medical Psilocybin member, and I am in charge of and chairing the education and training. Thank you.
Thank you. Lawrence, your mic is enabled. Please unmute yourself when you're ready.
This is Larry Lehman. I'm a member of the advisory board as well. area of expertise is research with psilocybin mushrooms, and I'm on the call to share perspectives regarding issues of dosage.
Thank you, Larry. James, your mic has been enabled. Please unmute yourself when you're ready to speak.
Thank you, sir. My name is James Brown, a doctor of pharmacy. I've been trained in psilocybin use in Oregon through a program called Emerald Valley Institute. I'm here today as an educator, as well as experts in dosing, administration, pharmacokinetics, dynamics, all that good stuff with pharmacy. It's a pleasure to be here. Thank you so much.
Thank you. Vincent, your mic has been enabled. Please unmute yourself when you're ready to speak.
Yeah, thank you. My name is Vince Espinoza. I'm actually an LCSW, so I'm licensed in Colorado, Oregon, and New Mexico. I'm actually pursuing Colorado facilitator license for natural medicine. I'm in a program with Fluence Training to get a certificate in psilocybin facilitation. So I'm on my 3rd out of 4 classes, and I will get my training license here in May to start my practicum in Colorado. So I'm here as a practitioner and a community member. I was born and raised in New Mexico, and so I have a strong interest in bringing this lovely medicine to our community and have witnessed firsthand how healing it can be. So, yeah, I'm excited to take part in this committee in any way that I can.
Thank you, Vince. Eileen, your microphone has been enabled. Please unmute yourself when you're ready to speak.
Hi, my name's Aileen Brewer, and unfortunately I'm sick. I'm losing my voice, so I hope you all can hear me okay.
Yes, we
Okay,
can.
perfect. So I am a researcher and I am a patient advocate. I mostly work with people who live with pain conditions, specifically headache diseases, but also other pain conditions. And I am excited to work with this community and hope that I can represent. Thanks.
Thank you, Eileen, and wish you better health. Dan, your mic is enabled. Please unmute yourself when you're ready to speak.
Hi, everybody. I'm Dan Jennings. I'm on the board as well, and I'm the chair of the Research and Continuous Improvement Committee, and I'm just here to be part of this discussion as well on dosage.
Thank you, Dan. Kate, your mic has been enabled. Please unmute yourself when you're ready to speak. Kate Hawk, your mic is enabled.
Hi. Yeah, it was actually disabled. Now it's good. Thank you, everybody. Good to see you. This is Kate Hawk from Santa Fe, and I've been involved with several committees, especially end of life care. But what's relevant today is probably my involvement with Scottsdale Research Institute and Sue Sisley in Arizona. I lived in Arizona. I've worked with her for several years, and I'm currently involved with their work with, they have a federal license for psilocybin mushrooms. So glad to be here.
Thank you, Kate. Gregory, your mic has been enabled. Please unmute yourself when you're ready to speak.
Sorry, multitasking and couldn't find the screen. Hi. My name is Gregory Evans. I am an independent researcher. I live in Santa Fe. I've been compiling research around analytics, genomics, phylogenomics, dosage, and how those dosages can be targeted towards specific therapies. Ian, I read your paper. I think that you did an incredible job. And I wanted to point out that, like, it's funny because a lot of the stuff that you wrote, I can tell you wrote because I know that it doesn't exist. And I've been writing the same stuff. So I can't wait to get into it and find all the parallels and find the next current version of truth.
Thank you, Gregory. Ian, that's all the hands. Let me just go through the chat. There is one person. My name is Ellen Schimmels. I'm a psychiatric nurse practitioner and Army veteran. I've been a mental health nurse for 28 years, trained in psychedelic assisted therapy through Integrative Psychiatry Institute. I'm also an educator. Happy to share my expertise as needed. Thank you. And Dominic has added some instructions in case you would like to speak. That's also on the screen in front of you, so whichever is best for you. And those are all. There's one more comment. Yeah, we came in. My name is Matthew Armstrong, Ojibwe descendant and psilocybin advocate. I do not have a medical background. Happy to serve, though. And that's everyone, Ian.
Perfect, thank you. If you can go ahead and go to the next slide, it will open the draft document that was sent out to everyone so that we can open discussion.
Ian, I'm just going to open it in a little bit of a different way. It doesn't show if I try to click on it and open it here, so I'll have that in just a moment. And just for those who are wondering, as we go through sections, I will be putting those into the chat as well. So just give me just a moment here. There we go. And can you see the screen at this time? Should be able to see the document.
Yes, so I'm going to go ahead and briefly go through this. That way we can kind of catch up and get where we are. So the psilocybin dosage definitions begins with something known as maximum theoretical psilocin. This is basically the amount of psilocin, the active ingredient in magic mushrooms, that is systemically available to our bodies. Now, I do want to note this measurement is using a stoichiometric conversion. That means that sometimes what happens in chemistry labs is not exactly what happens in our bodies, and that's okay, but we are going to use what we have the most evidence on, and that of course is going to be our stoichiometry until we can collect more through this program and through efficacy. So the MTP calculation is just measured amount of psilocin plus the measured amount of psilocybin with a stoichiometric conversion factor. That allows us to see the maximum potential amount of psilocin that we are going to be dosing someone. It allows us to have one consistent unit of, hey, we're going to prescribe this amount or this range. Here's what we are listing within the range, because we have two pharmacokinetically different molecules that we are working with within the mushroom itself. So going into that further, we then have something known as an available psilocin fraction. So this describes the amount of psilocin relative to the total measure of milligrams of psilocin equivalents. So it is our milligrams of psilocin over MTP. That's going to give us a percentage of pure psilocin, active psilocin, that already exists. So some things we know about the pharmacokinetics of psilocin. There's not a whole lot of data, but we do have an understanding that it is much faster because it doesn't have to go through the bioconversion into an active molecule. So what we use this APF for is to say what percentage is psilocin, and that way when our facilitators are going to be administering this drug, they can say, hey, we know this has X amount of faster acting medicine, and we have Y amount of slower acting. That will allow us to not exactly predict, but it will give us a frame of reference for the medications that we are providing to these patients. So a higher APF, greater proportions of immediately available psilocin are associated with higher likelihood of early onset and more prominent early psychoactive effects. Lower APF indicates a greater proportion of psilocybin, requiring metabolic conversion and are associated with a higher likelihood of delayed onset and extended duration. So we are going to be trying to provide as much information to our facilitators as possible in order to ensure that they can do their job correctly. So the next thing we go over, and this is the most provisional part, is something known as onset profile categories. So these onset profile categories, each is just a measure of how much the percentage of active psilocin impacts the onset of action. So we know that psilocin has a faster onset of action. We don't have specific numbers on that yet. There isn't extensive research on the pharmacokinetic differences between psilocin and psilocybin. Therefore, these recommendations are provisional. We recommend their use for clinical planning and monitoring. They are not determinative classifications, and thresholds should be revisited as outcome data accumulates. So you'll see later that we forward a lot of this to the research and continuous improvement arm, because I do believe that there is certain evidence that we don't have a lot of at the moment, like the effects of psilocin, how psilocin and psilocybin produce different systemic effects. The next thing that we have are just dosage levels. This would allow us to provide a recommendation. So if a practitioner is going to provide a recommendation, they can say, okay, I want you to have a subperceptual dose for, let's say, as an onboarding ramp, right? We know patients with PTSD may have a higher incidence of what are known as bad trips. Therefore, a subperceptual dose would allow them to kind of see the lay of the land before we go ahead and give them a therapeutic dose. It's not meant to be something that is, totally provided. There is not enough evidence at this time to meaningfully say that this is safe and effective and this will work for these conditions, but it can be used as an onboarding ramp, so I didn't want to disinclude it entirely. A perceptual dose is a dose that is expected to produce noticeable perceptual, emotional, or cognitive effects. So we have 3 tiers of this, or 2 tiers, sorry. We have a mild perceptual, which is basically above a subperceptual dose, but below 1 gram of mushrooms in equivalent, so 10 milligrams of MTP. We have a therapeutic perceptual, which is going to go from 10 to 30. And then we have later a high dose, which is going to give us the ability to recommend these higher doses that we don't have as much, evidence behind for certain people. Maybe they had a previous experience. Maybe they are on medications that are going to blunt the effects of psilocybin. And so this gives practitioners an avenue out to say, okay, we understand that everyone has different circumstances, and we are going to ensure that we are not blocking anyone via overly stringent dosage regulations. I will now open the floor to any discussion that people have. What questions, what things can we change? This is a first draft, so I want to see what the committee thinks.
Chris, first in line, you may begin.
So I'm not sure I agree with using total psilocin content. I think a better standard dosing might be total equivalent psilocybin. I think this is going to create a lot of confusion for people. And so that's like the first comment I got on that. Second is, as we're going down and looking at the subperceptual doses, I think most people that microdose actually perceive perception changes. And so I don't like that terminology. I think subhallucinogenic might be a better terminology in that regard. But those are the first couple things that stick out to me.
Thank you, Chris. Larry, you are next in line.
Yeah, thank you. And thank you, Ian, for the document here. I think we're getting ahead of the evidence and the science here, and I've not seen the APF used in any articles. We actually obtain psilocybin from the Scottsdale Research Institute. It's approved by the FDA. They're not doing these type of calculations. My own research is that the psilocin can be highly variable based on which flush you have, environmental conditions. The actual absorption can also be varied by the diet and different things. And I just think we're getting ahead of the information, and I would propose not to include the APF. I'm also concerned that we're maybe producing an undue burden on the growers if they're going to need to, recheck every flush. So that would be my concerns about the APF. The perceptual dosing is a challenging area. There's almost no evidence really regarding the role. It's quite common in the community and what roles are there. And I'm not sure there's a reason for us to get into making definitions and actually suggesting which type of product should be used for something that we don't have any evidence for its benefit or lack of benefits. I just feel like in both of these areas we're getting ahead of the evidence, and it would be best not to include.
Thank you. I do want to challenge back on the APF. Most FDA-approved studies are only going to use synthetic psilocybin. Therefore, they would not be working with the biomass that we are currently working with as a program. In that sense, we have to take differential actions from what the FDA is doing. Since we are a program that is allowing the natural medicine, we specifically exclude synthetic psilocybin and psilocin. We have to take into consideration the fact that these plants or these fungi are going to grow with psilocin and psilocybin, and we need to be able to inform the patients and the facilitators what percentage of what are they being provided. Anyway, in regards to that was my only response. That's the clear differentiation between a lot of the studies and a lot of what we have here. And so I did want to clarify on that point. I will go ahead and turn back to more public comment.
Thank you both. Gregory, your mic has been enabled. Please unmute yourself when you're ready.
Would you believe that I've already written up a response and have an entire document to submit that I've just finishing now, and I don't have time to get it in, and it's going to be a whole thing, but I think this conversation will be great. I want to be super clear. I could talk for about 20 minutes in response to this, and I want to be respectful and constrain that. So yeah, I just wanted to get consent to begin there before because I don't want to have 5 things to address and didn't want to speak over everybody.
So Greg, are you asking that you want 20 minutes? Is that what you're trying to say? I would say if you could limit what you have just so that we can, do this. And remember, this is a beginning. As Ian said, this is a very rough draft. We're going to have many more conversations on this. So what I would say is if you can limit it to 5 minutes, as we've kind of, oh, I'm sorry. Did we say 3 minutes on in the committees? My apologies, I forget off the top of my head.
It's 5 for the committees because there is a smaller population.
Gotcha. I couldn't remember if we went 3 and 5 or which way. My apologies. So Greg, if you could just limit it to 5 minutes, that would be great, just to make sure we hear everybody. And so I would say pick your most important aspects. But thank you for asking.
Yeah, in that understanding, is there a potential for me to screen share?
I do not have a way to turn that over to you at this point. So yeah, but you can, as always, you can send the documents with context afterwards and we can put it in the meeting record.
Yeah, I think I can actually, there's a graph I want to present too. There's a chart and a graph, and I can publish those and provide a link and put that in the chat if it's okay. I'll do that after I speak.
That would be fine.
Okay, so I have an assumption of where you extracted the acronyms from. Probably the total potential psilocin from Oregon's 333.064 0150 section 50. I have a recommendation that we shift away from using the term theoretical and move towards potential psilocin. I think it's more accurate and it gives us the ability to aim to a target. So the construct that I would like to present is the acronym would be PCN, and this stands for psilocin normalization equivalency. And so that directly works with total psilocybin. So I put the psilocybin equivalency of that and call that PBCE. And those can be easily calculated from one to the other. I am on the side of the fence arguing that we should go as for labeling, so testing, getting the analytics and labeling, we have an opportunity to push this to a medical market and shift the perspective to understand that we should be measuring psilocin equivalency. Several companies that do analytics already utilize the concept of psilocin normalization. Tritomics, Light Labs, I don't know, we'll ask whoever else is in the room from the analytics community. Moving on, key target dosage ranges. Microdose should be 0.1 to 0.4 milligrams. This is a subperceptual, no acute effects. I have what I call a calibration dose, that is 0.5 to 1.5 milligrams psilocin equivalents. So all of these measurements are in PCN, psilocin equivalent normalization. The calibration range is kind of what you're looking for when you have this like finding the threshold in a non-threatening manner. This is a technique that I think could be really useful for people in the future. I also don't want to get into what Larry was talking about, like over specifying this and making recommendations to the way people should present their therapy. Like I want to keep it into clinical definitions, right? So moving on, we have threshold at 1.5 milligrams. That is your first perceptual dose. There is a range that shifts, but that is like the average target. A mini dose, which colloquially would be your museum dose, that's 2 to 4 milligrams. You feel it in your body, mild perceptual shift. Pre-therapeutic, that is your 3 to 7 milligram range. That is the transition between non-therapeutic to therapeutic. Therapeutic range is 7 to 18 milligrams psilocin equivalency. That 18 milligrams psilocin equivalency is your 25 milligram psilocybin equivalent.
Greg, you have 30 seconds.
That is about it. I have a whole conversation about the interaction between psilocin and psilocybin. I think we should probably take that out for now, and reframe some of that. I yield.
Thank you.
very
And
much.
I always read the public comments after every meeting, so please feel free to submit it to the medical.psilocybin at, what is it, Dominic?
It's medical.psilocybin at doh.nm.gov, and I will be putting how to submit written public comments in the chat momentarily.
Yeah, I do appreciate all of the work that you have looked into this. And then we can go ahead and move on to the next speaker. Thank you.
Eileen, your mic has been enabled. Please unmute yourself when you're ready to speak.
Thank you. I just wanted to respond to the gentleman who said that he was unsure if we should be addressing lower doses and microdoses because the evidence isn't there. While I recognize that the initial targets for this bill are not pain care, and that may be outside of the scope of the medical rollout initially, I do know that one of the main goals is to allow for end-of-life care. And end-of-life care includes pain care. What we know right now about pain care with psilocybin is that it includes smaller doses more frequently administered than would be for, say, depression or anxiety or PTSD. And because we know this, I think that it's important to continue to consider those lower doses and how they can be included without saying how often they should be administered, because I think that's really up to the practitioner and the patient that's in front of them. I also think that we might want to consider a different word than therapeutic for the perceptual doses, because therapy can come for many different conditions in many different ways and at many different doses. Thanks.
Thank you. Eric, your mic has been enabled. Please unmute yourself when you're ready to speak.
Hello. Just wanted to say that just for some of the people that are, wanting to not have the regulation or the guidelines stating how on dosage, I would say that the reason we need this laid out is because things get lost in translation. Not everybody's going to be growing the same strains or fruit bodies in potency. If you are, able to acquire genetics for a certain species or for mushroom, for a certain, cultivar, and you're running that in your facility, it doesn't mean that the next facility is going to be running that same strain or species. And for the, uneducated individual out there, they, would be under the impression they're going to be having, a certain experience with a certain amount of one, and if they're under the impression that it's equal across the board, then there's going to be issues with that. And also some of these dosages that I'm seeing in the document, I don't know if anyone here has personally experienced a 1 milligram dose of psilocin, or even up to 3. I think it was 1.5 that it said in there. That's quite a strong dose. So that I currently have experience with 3 milligram gummies. That is pure psilocin extract, or or the psilocybin equivalent, and those are above social experience. Basically, like you can't drive, you can't do things like that. So even that metric of measurement is quite high, in my opinion. So it's we do lose things. We do lose the mean. Things get lost in translation when we start going into these specific measurements. And I just wanted to give that input. And if anyone has experience with the psilocin extract, they could probably add to that. So just so just psilocin as a measurement. There's other alkaloids that cause different things, and so I got a guideline of psilocin measurement. We are, we're kind of missing some of these properties that are going to have an effect on the experience. So that's all. Thank you.
Thank you. James, your mic has been enabled. Please unmute yourself when you're ready to speak.
Thank you, everyone. I think you guys had a key point earlier. This is initial discussions. I think we need to keep that in mind as we go through this. Ian, you've done a great job at just presenting something to us. I do appreciate that. I'm on the side of Larry's fence, listening to him today. I want to just echo the same similar things. I don't want to have to repeat it, but I just definitely want to say, echoing Larry's, I think this complicates things, and I know we have a year to do this, and I just want to make sure we make this easy enough for people, but there's enough information here that's very important. I am in agreement on the milligrams. I think that's a standard that we've seen across the board in the literature. This discussion between psilocin and psilocybin is very important. I think that also involves a lot of the enzymatic processes that are due to that change, unless we're just giving them the pure psilocin conversion. And then again, if you do give somebody pure psilocin, you have that quick onset and shorter duration. So we got to look at that stuff when we're talking like this, Ian and group. But great start. I do have some differences on the dosings, of course, but I think as we get through that, I think that will be maybe a little bit of tweaking, stuff like that. And then the wording, I'm glad somebody brought that up too. I just didn't know if that therapeutic word was something I had caught too. I don't know if we want to use that, but it is the medicine. So therapy does make sense on that word. And I think that it's important that we also sit here when we do look at these type of proposals like this, and look at the individual. When we're thinking about this, we need to think individually. Not everyone's going to have the same experience. Not everybody's going to break down the same way. Not everybody's going to do this. And so there needs to be, I'm not saying flexibility, but just thought process behind our decision making at the finals. Is that I just want to make sure everybody's aware of that consciousness of this is individuals here, and it's sometimes there needs to be a little flex or thought about that. Thank you so much.
Thank you.
Thank you, James. And to kind of agree with you, a lot of these dosage ranges, and I'm sure anyone that's read the literature, they are all over the place. They go from 0 to 100, and it is often difficult to say with certainty where the minimum effective dose is or where the therapeutic range is. So I am fully open to changing those milligram proponents. Something that I am concerned about is losing the actual natural medicine component of it. We are working with two chemically distinct molecules, and so it's very difficult to say, hey, we're just going to label this with the amount of psilocybin that's in it and not the amount of psilocin. So the most of what this was to do was just to give us a framework and an ability to collect more data, because right now there isn't enough data on how psilocin is converted in the body. There's a good amount of data on it from a chemistry standpoint, just not about a biology standpoint. And we all have our internal mechanisms in biology that is going to contribute to how these things play out. So I would be more than happy to strike the interpretation and clinical significance of APF. But I do think it's important to, inform our facilitators, hey, you're giving someone a very big dose of the immediately active psilocin pursuant to whatever strain that grew that way. And I think they should be, warned and said, hey, like this may kick in faster. You may have a shorter duration of action. You may have some other factors that are going to play into this. What this originally was meant to do was just to say, hey, here's a way that we can calculate the amount of psychoactivity, if you will, in this medicine, because ultimately there are two competing chemicals in there. The reason why I chose psilocin instead of psilocybin for the basis of this dosage framework, to answer some, that psilocin is the active one. It's the one that's actually producing the effects, and therefore it makes more sense to measure it in the active ingredient, what it's going to turn into, than try to imagine how to backtrack a chemical reaction that has already taken place and how much would be there. I just wanted to clarify those points. For the dosage ranges, though, I am more than open to changing those, swapping them out, whatever we have to do, because I have said this from the beginning. There is a lot more knowledge in the community in the state of New Mexico than is in just my brain. So whatever things we need change, we can do. I'm really happy for this discussion.
Thank you, Ian. We have comments in the chat and 2 hands raised at the moment, and I will start with the chat. Shane McDaniel, will there be a difference in potency within the same cultivar, for example, Golden Teacher, between different cultivators depending on grow conditions and other factors? Question mark. Kate Hawk, I appreciate the recognition of the complexities and nuances. I applaud efforts to give maximum meaningful information while preserving flexibility for appropriately dosing people with unusual tolerance or other conditions. Gregory has a response to Shane McDaniel saying, potentially, possibly, likely, this is a great conversion for propagation. And now we have a hand up, unless you wanted to answer any of those or address those again. I mean, no. Okay. We'll go.
Yeah, I can kind of go ahead and it's kind of like in the Medical Cannabis Advisory Board, they defined a unit of THC being just 200 milligrams of, or a medical cannabis unit as 200 milligrams of THC. And so they did forget some of the other chemicals. So I do recognize where they are saying, you know, alkaloids and other chemicals that can be part of it. But in terms of what we have as defensively psychoactive and are the usual culprits of what produces a psychoactive experience, that's what we're kind of working with. I didn't set any type of here's 1 unit equals this. I just said, hey, if we took our psilocin that's in this mushroom and we took the amount that we could stoichiometrically get from psilocybin, then we would be able to have a dosage definition that would work across mushrooms even without, you know, without having to have 2 dosage frameworks, one for psilocybin and one for psilocin when they're both growing in the same plant or fungi. My mistake. I'll go ahead and turn it again to public comment.
Thank you. Adrian, your mic has been enabled. Please unmute yourself when you're ready to speak.
Hi, thank you. Yeah, I'm Adrian, owner of Druid's Choice. I'm currently a cultivator in Colorado. I do want to comment real quick on the average doses that are being sold here or utilized here in Colorado. No one is really interested in microdosing here, mainly because the regulations say that the patient has to stay at the facility for 2 hours after a microdose. Now, those of us that have microdosed personally, we know that you don't need to stay there for 2 hours to microdose, but that's the main reason why we're not microdosing here. But there is a benefit in microdosing for certain conditions. Number 2, on the comment that Shane McDaniel made, as far as difference in potencies, there's huge difference in potencies for the same. For example, Golden Teacher, my current harvest is at 4.7 MTP. But I've seen some from the other cultivators as low as 3.2, 3.5 MTP, all Golden Teacher with the same alkaloid background or the same present alkaloids, you know, so to confirm that they are all Golden Teacher. And another thing on your APF, that's not something we're doing here in Colorado, but I do know that, you know, higher psilocin in the fruit is a quicker onset and more body load. Higher psilocybin is a more extended duration of the effects. And then the standard dose here in Colorado has been about 30 milligrams. I do sell mainly 30 milligrams with 5 milligram boosters or 10 milligram boosters to take that 30 to 35 or that 30 to 40. When I first started a few months ago, people were requesting 20 milligrams, but I guess everyone's averaging out to higher, between 30 and 40 for standard doses, and really nothing less than that has been requested from me specifically. And that's all my comment for now. Thank you.
Thank you. Chris, you are next in line.
You know, I just wanted to say, I understand why you're going with, you're thinking we should go with psilocin. But again, I think it's just going to create a ton of confusion because most people don't know what that is. They're going to be looking for psilocybin content. People have been knowing and engaging with this plant as psilocybin for a long time. So I'll make that case again to use a different equation to look for total equivalent psilocybin. And then as far as the rest goes, is like quick onset and everything, there's so much patient variability and people have different sensitivities that dosage is going to have to be highly individualized and regardless of whether you have, a large amount of psilocin available immediately, like somebody could have an early onset without that. And from a facilitation standpoint, it doesn't matter. Like you're there to support them through that journey, regardless. So thank you.
Thank you, Chris.
Thank you. But I do think it is incredibly important that people can be educated on what they're getting themselves into. If we're not providing them the proper education on what they are taking, then we can't have meaningful, informed consent. And the reason that we're doing this again is because it's a lot harder to say, okay, how much psilocybin is from psilocin than the other way around. We could stoichiometrically reverse it, but that wouldn't be applicable to how the conversion actually happens in the world. But I am very concerned with making sure that we can provide proper informed consent to patients because they need to know what is happening in the body versus what is just going to hit them with a quite rapid onset. I will go ahead and turn to Larry. He had his hand up.
Correct.
Thank you, Ian. And just one more comment. And I totally agree with you. Almost all the research has been with synthesized, but we are actually using the whole mushroom here and working with Dr. Sicily. And what the FDA has mandated is actually psilocybin is what they're keeping track of, and that the psilocybin needs to be plus or minus 10% of what's being described. I think part of that may be the difficulty of measuring and variations in each batch in different flushes. So I really hope that whatever we develop is something that won't be a burden to the propagators and won't confuse the facilitators. Thank you very much.
Thank you so much. And I do appreciate your comments because in all reality, and this may be an issue more for propagation and manufacturing, but psilocin is a lot less chemically stable. So even if how long are those testing requirements going to last for and things like that can also play a factor. I am not going to claim that that draft that I provided you guys was perfect and needs no correction, but I do say that it does provide at least some framework that we could hash out the details because I don't know everything, unfortunately. I think it would be kind of cool, but I do really appreciate everyone's comments, even if you're feeling like it's being contrarian or anything, this is inherently a process of saying what is going to be best for the patient, and I appreciate all questioning from anyone. I'll go ahead and turn it back.
Thank you. James, your mic has been enabled. Unmute yourself when you're ready to speak.
Ian and everyone, I really like the idea of the psilocin, and I like the idea of the psilocybin because that's what's currently being trained on and being educated around. So it would be our way to include both because we are setting new, as they would say in Star Trek, right? Like new frontiers. We're doing that right now. And sometimes we need to think outside the box and say, maybe we need both. At the end of the day, just a thought, because We can train on both. We can educate the public on both. It's going to be clear. We can really dial that up and say, here's your product. It's kind of like the cannabis and CBD. You get so much THC, you get so much CBD. You're getting so much psilocin, you're getting so much psilocybin. Maybe you don't get any psilocybin, you just get all psilocin. That'll be up to the therapy plan. And that's my next point. Some of these dosings are all going to be based off these therapy plans for individuals, based off of that relationship that's created. And so just wanted to think about that too, is we're going to back into some of this stuff as we get the therapy plans together for the different, ICD-10 wonderful things that we've done so far. So that's all, Ian. That's my comment on that. Thank you.
Thank you. Dominic, I'm going to let you go ahead in line if there's something you want to address.
Sure. Thank you, Jorge. And just so everybody knows, this is a great conversation and I'm really glad it's happening. Just to kind of address the issue and why it's so important to have this now is when we did create the units with regard to cannabis, part of the issue, it wasn't so much that it was forgotten about the other aspects, it's that the testing wasn't very reliable yet to be able to actually measure them very well. And so when the unit was created and it was decided to be based just on THC and not the other potential, items that could affect that entourage effect, it ended up being there. And so to change it years later, when the measurements were going to be much more precise and able to measure other forms and other aspects, CBD and everything else, became very difficult to do because the industry standard had already been set at the THC. So the compromise made was to ensure that things were included on the label, but that the measurement with regard to units stayed at the THC. And so I bring that up because it highlights why this conversation right now is so important and why I can say it's not going to be the last time we have the conversation, because we have to make sure that we are able to put this into place in a way that it's going to be beneficial, not so restrictive, but also that we could potentially change it down the road as the research continues and we find out the things we do not know at all right now. And so that's just one of the things to think about. We know a fair amount of things regarding psilocybin and psilocin, but there are a lot of things we do not yet know. So we have to think about that when we look at regulations and protocols and dosing. So I'm just going to leave it at that. It's just great conversation and I'm really glad that it's happening. So thank you.
Thank you, Dominic. Anne, your mic has been enabled. Please unmute yourself and when ready to speak.
Hi, everyone. My name is Dr. Ann Metz, and I'm a licensed professional counselor, and I'm also a natural medicine facilitator. I'm licensed in Colorado, but I live in Taos. And I would just ask for a purely pragmatic reason to stick with psilocin, since that's what Colorado has done. I know that, we're doing a very different program, but just for sort of practicality for facilitators, the way I do informed consent, the way I talk about dosing in the non-directive model in Colorado is in psilocin. And so from a pragmatic standpoint, it would be lovely to be able to continue to talk about it that way in New Mexico. And I imagine that lots of facilitators will be licensed in both states. And so I suspect I'm not the only person who might bump up against that. But thanks for all your work and great document, Ian.
Thank you.
Thank you. Shane, your mic has been enabled. Please unmute yourself when you're ready to speak.
Yeah, I'm maybe in the minority here, but I'm going to take a chance on it that what I have to say might be valid. The ancestral users of this medicine have been using it for hundreds, if not thousands of years. They do not dive into the weeds like we're diving into here. I'm talking about psilocin and grams and micrograms and psilocybin and tryptophans and everything else we're talking about. I understand the this is new, and we're trying to dot all our I's and cross all our T's, but we can get into this position where we're in a paralysis by analysis, and we can't get anything done because we are analyzing things so far down into the weeds that it becomes counterproductive. When, if we're looking at patient safety, from my understanding, the harmful dose or the lethal dose is so high that we'll never have anybody reach the harmful or fatal dose or lethal dose. We might have some trial and error periods with certain patients where, we might start off with too low of a dose and then have to go up, which means that patient is experience double cost because they have to go back for a new dose that they didn't get right on the first time. I just think that, like I said, the ancestral users of this at most would measure it out in dried mushrooms, not in the different chemical compounds or the different strains or all that. And again, I'm not saying that we shouldn't pay attention to this. I just don't want to get so deep in the weeds on this that we forget or we get, to a point of paralysis by analysis, if that makes sense. Thank you.
Thank you, Shane. Denali, your mic has been enabled. Please unmute yourself when you're ready to speak.
Hi, everyone. I'm really appreciating this dialogue. This is definitely not anywhere close to an area where I have expertise to offer, but I wanted to offer a couple of process recommendations or questions. I think it would be helpful for me at least to hear maybe from the department what the most important pieces of dosage are for the program to be able to move forward. Like, what is the minimum requirement for regulation that the department needs, from the department's perspective, needs to be in place for this to move forward? And then I would recommend sticking as closely to what those minimum requirements are, just because it feels like the consensus here is that we're learning so much, and we will, as the program enrolls, continue to learn. And so if we can regulate as leanly as possible, it makes it a lot easier to adapt and move forward. And I just want to recommend too, I know that there will be more conversations. I think it would be especially valuable to have the input of the healthcare authority member on the advisory board have input here. I know I'm joining from the Roundhouse, and I keep seeing them around here too, so I know they're very busy with session, but I hope that we won't move forward without hearing from them their input from both a patient safety perspective and what is going to be possible for coverage and reimbursement, and what data points they're going to require when we're making dosing decisions. They'll just have some unique perspectives that I think need to be captured and want to be sure as a part of the drafting.
Thank you. Ian, I just want to catch up on a few things in the chat, and then before we go to the next hand, let's see where we were. Gregory dropped a link in the chat as far as propagation, and then Eric says, yes, Gregory, I would agree with your dosage recommendations, especially in the microdose levels, 0.1 milligram as opposed to 1 milligram psilocin, much more practical posed. Gregory added, can we get Barry Dunn or Scott Folkman to weigh in on the commonality of using psilocin normalization in the industry? And then Eric says, there needs to be an improved universal metric. I vote psilocin as the most comprehensive metric. Psilocin EQ is is that metric? Paul Walton, it would be great to see a microdosing program, maybe a standalone program. Amongst others, our veterans and first responders are taking pharmaceutical depression and anxiety medications. Microdosing can get people off of these pharmaceutical medications. Additionally, some veterans and first responders are going back to work and receiving more trauma where microdosing can help with some of the negative side effects. Treating microdosing as other medications that allow people to be adults and take responsibility for their medication treatment and to not take more than prescribed, that would lead to impairment. Thanks. Laura added, I agree with Shane, and Greg added in the chat, the way I see authorizing microdosing happening is an iterative one if we write the language now, but the framework for where it could exist. As we roll out the program and collect data, we will absolutely be able to collect the data on potency analysis to help build the effectiveness for safe use. And now we have a couple of hands up. Greg, your mic has been enabled. Please unmute yourself when you're ready to speak.
I wanted to kind of address some of that stuff about traditional use. And I come from entering this space by like counting mushrooms that I picked from the ground. Traditional use is commonly, traditional ancestral use is commonly, you know, you have, I mean, if you look at Sabina's work, it's like you send the kids to the forest to collect a number of mushrooms during a certain time of day, and they walk back and they avoid certain omens, and they bring these to the house, and then you distribute pairs of certain types of mushrooms. Other types of mushrooms you separate and consider in different different ways, right? And there's all complicated spiritual framework for this. I don't want to take that away. I don't think there's any world in which the spirit and the intuition behind dosage can go away. Like when it comes to the end of the day, like I'm the one that's counting this the most in the room. But when it comes to me taking mushrooms, I've listened to the mushroom, right? What we're trying to do is create a framework that will influence the people in the seats of the medical industry, that we have done our due diligence, that we understand what we're recommending to people, what we're asking people to do, we're asking people to understand. So if I'm a facilitator and I am a person that likes to do a muscle test when I'm choosing a dosage for my client, I have an amount of mushrooms that I know fits inside this box. But then the process and the ritual of helping that person be a part of that decision, nobody's trying to take that away. This is not about telling facilitators how much to give somebody. This is about understanding what the terms are, how somebody in a lab somewhere can understand something so that we can really provide true harm reduction. Because if you don't understand how variable mushrooms can be, and you don't know what the variability is in that mushroom, most of the facilitators aren't going to be providing or growing or testing their own mushrooms. It's very important that they understand that. I do think that the idea of putting the alkaloid ratios between psilocin and psilocybin on the package is a good one. You can easily put PCBE on there right next to PCN, or however you want to acronym that. Put the psilocin and the psilocybin. It's a simple math equation. And then one typo that I found in your document, Ian, was the 30 milligrams that you calculated from 25. You calculated that the wrong way. It should be 18 for the high dose. But yeah, nobody's trying to take the spirit away. At least I'm not.
Thank you. Yeah, and in all fairness, I do want to point out that it is one document, but it's really two documents in the sense there's a definitions and then there's a dosage levels. So we can kind of separate those two ideas because I think they are not pointing toward the same thing. Personally, I am not a facilitator in Colorado like a lot of you are. So seeing those numbers that you guys see all the time is not something that I do. So I really do welcome all input from wherever you can bring it because it is important that we don't take the spirit away from this medicine. And it's also important that we ensure that if we are going to put whenever a medical provider makes a recommendation, they're putting their license on the line. And so to make sure that they are comfortable enough saying, here, I recommend this is a really big part because the legislation that was passed requires a provider authorization. And so we need to make sure that they can see this and not just immediately make it illegal by proxy because we didn't set up a robust enough system where we can ensure patient safety, where we can ensure that our dosage levels are correct, where we can show our data that we are going to collect. So I think this conversation is a really important one, and I do appreciate all comments. So I believe I see James Brown with his hand up.
James, your mic has been enabled. Please unmute yourself if you're ready to speak.
Thank you, sir. Appreciate you. Ian, just a thought too on as we get to the different dosage levels or different regimens, can the thought of a split dose so that let's say the total dose, there's need to be any type of caveat or sentence that says that 30 can be split into two. They would receive the total 30, but it gives that safety profile a little bit more buffer there if that's needed between the two. Makes sense? So when you're there and you're in that session with that person, and if you feel like they changed or something and their safety plan allows that again, because we won't go back to safety plans, if that safety plan allows this, then you could split it. But that has to be kind of written in the original therapeutic regimens. I just thought that, I don't know, what's your thought on that? If we have some type of terminology added that says that the higher doses may be split, but the patient still would receive, based off the therapy, they might only use half. I mean, it depends on what's going on, but I just wanted to see if that was available as an option.
In my head, yes, that was kind of how I imagined it. It's why there are, there's no exclusion for how they have to be spread apart. I really wanted them to start low and slow, right? Kind of like we tell people for cannabis, low and slow, because if once we have a bad reaction, it's a lot harder to reverse that bad reaction than if they're starting to see paranoia, we can use our therapeutic techniques and just kind of stabilize them down, but we're not going to continue to up the dose of a medicine that could be causing paranoia. So I do believe that would be a good idea. And then in future drafting, we will include that. This is not the final version. This was something for y'all's brains to digest and to say, I don't like that, or I do like that part, or anything like that. I really appreciate you guys.
Okay, we have a couple of comments in the chat. Deb Thorne, functional kinesiology equals muscle testing for a substance. And Gregory Evans added, I would like loudly not put any regulatory language on dictating max dosage.
In the current dosage levels, there is no regulatory maximum for dosage.
And then Eric Baca says, maybe we leave dosing to open and just come up with guidelines for testing for known tryptamine content. And we have a hand up. Greg, your mic has been enabled. Please unmute yourself when you're ready.
Yeah, feel free to cut me off if this is out of scope for this conversation. But just experientially, like through my journey, as I began cultivating and did not have access to HPLC or mass spec data, or even now that I do, I just don't have the the money as an individual to send off a chunk of what little I have. When I do get data, it's really valuable for me to walk it back against my experiential data. But if you look at the chart I posted, I have this little calibration zone. And essentially what I'll do when I get a new batch is I will eat a very little bit of it. And then I will eat a little bit more. And then I will eat a little bit more until I can just feel it. That's like the eyes start to wake up, the tingling happens, the early sensations just start to let you know that it's there. That's how I find my target for that species, that cultivar, that batch of how much I want to use for a microdose or how much I want to use for a macrodose. Each person is going to find their calibration a little bit differently. So there's a range in there, and it's like flexes in and out of that. But there is a very safe process of titrating into a known first interaction with the molecule. I don't think that that's something that people necessarily need to do with every patient. And if we understand that math, and then we can apply analytics to sidestep some of that, because like most of our facilitators are doctors, they're not going to be eating those mushrooms for a week before they hand them to somebody. Like they need to walk in there with a known product that has been just understood and handed off. There's a lot of this stuff that really belongs in the propagation and analytics side of things, and we'll talk about that in 20-30 minutes or whenever that happens. But yeah,
Thank you.
I mean, I don't know if that's too much for here, but I mean, yeah, that's my take.
It is a good insight into how dosage can vary. I think it almost mirrors the entourage effect a little bit, in that different alkaloids can provide that, but a lot of that would be for dosage and them. The
Yeah.
department had a comment.
Oh, can I plug one thing before I have to talk again?
Sure.
So like, I want to test for all alkaloids and develop, I have this concept of a fingerprint. So every cultivar is going to have like a unique effect, right? So we have to normalize the PCN dosage to begin to understand that. So the alkaloid ratio between psilocin and psilocybin is only one aspect. And I think that's enough to put on like the dosage kind of calendar. But then everything outside of that, how every part of that mushroom affects it, is like this fully understudied branch of research that like I am very interested in. And nowhere in the world, nowhere in history have we had the opportunity to get that information. I just think that we can, and I look forward to understanding these mushrooms a little bit more.
So yeah, the testing would definitely be a conversation for the dosage or for the propagation committee. But in terms of testing for all alkaloids, I wouldn't have a problem with that. The reason I base the dosage as of now on psilocybin and psilocin was that those are the two alkaloids that are specifically laid out in SB 219, that the law that we're operating under. So I just went with that, being that that was the natural regulatory step, was to regulate those two substances specifically. But I will go ahead and turn over to Dominic.
Yeah, and you're not wrong, but the market will drive it.
Thank you, Ian.
Absolutely.
And I just want to clarify something is I think some people have the misperception that everything that we're talking about here within this committee is going into the regulations. And Denali had that question about what is needed by the department with regard to the regulations. A lot of these ideas around the dosage and the administration are going to be a part of the protocols and the guidelines, not necessarily a part of the regulations. There will be some aspects that will be in regulation. For example, what is going to be the definition of, how do we measure? So that is definitely something that needs to be discussed, but that does fit a little more in with the propagation and testing. But either way, most of what we're talking about here within this aspect is going to be a part of those recommendations, guidelines with regard to it. And part of the reason for that is, as a few people have sort of noted in the chat, we don't want to put an actual dosage into the regulations because, again, as the research comes out and we're able to fine tune things a little bit, and as individual patients have different levels that are going to be appropriate for them, we don't want to hinder the process, and that working together between the medical and the therapeutic practitioner and the patient. And so just remember those aspects. A lot of what we're talking about here are going to go into those guidelines and protocols, not necessarily into the regulations. So I just wanted to make sure that's clear so that people know this is an ongoing and evolving area of it, and we're going to do our best not to lock it in so that we don't recreate, as we mentioned before, that mistake of suddenly only having one item, one aspect of it that is actually creating problems and issues down the road when we have more information in 2, 3, 5, 10 years. So please just do remember that. So if you're worried that, oh, we're suddenly going to have all of these doses in the regulations, that is not the intent. Anyway, back to you, Ian.
Yes, and that's another reason that I provided ranges, is because what a lot of studies are using weight-based calculations of dosages. There is some controversy about whether that is applicable with psilocybin. But when I provided the original dosage, subperceptual dose of 2.5 milligrams, it wasn't thinking of someone, 90 pounds soaking wet with rain boots on. It was someone that is, maybe larger or has more body mass. So that maybe it takes more for them to get a subperceptual dose. While the information is still evolving, we have an obligation to collect data and to ensure that we know what we are giving patients. So I believe at least the first part of this, the MTP and APF, are pretty solid in what they do. And I haven't heard a whole lot of objection. I've heard some about the APF, but can we all get an agreement about MTP? Is that something we can all agree on? I would love to hear your comments on that. But I will turn back to Jorge.
Okay, we do have one hand up, but I want to catch up here in the chat. Let's see, Eric. Let's see. Trying to find out where I left off. Here we go. James Brown added, I am in favor of having both the psilocin and psilocybin being reported for increase in analysis and enhanced public safety. Eric Baca says, yet to be determined. Scott Folkman, testing should probably be done for psilocybin, psilocin, baeocystin, oh, here we go, norbaeocystin, and arugasis. Oops, I'm not sure if I pronounced that correctly, but here we go. That's in the minutes. They all have different pharmaceutical kinetics and potencies. Further research is needed for better understanding of their independent and entourage effects. But if the concentration of each of these is known and reported for each batch, they can each be reported, which prevents some confusion with the definitions. Gregory Evans says, I would love to have access to vagal tone measure and gut health prior to and after dosage to define the human elements of the equation. And Kate adds, more appreciation for the sensible, well-informed process. There are several lenses that we're looking through: scientific, therapeutic, public perception, lawmakers, observations from groups in Colorado. With the same batch of mushrooms, some participants with apparently similar bodies had roughly half the effect on twice the dose. And then James added, I agree with MTP definition of available psilocin fraction, add definition of available psilocybin fraction. I also agree on excluding the others for entourage effect if it's cost effective. I can go back to raised hands here. Shane, your mic has been enabled. Please unmute yourself when you're ready to speak.
Yeah, a couple of things. We've had a lot of talk on keeping price down. the more requirements we have on laboratory testing and what we want them to test for. And again, I go back to, I understand the need to be safe, but are we being so safe that we're making it so expensive that we're pricing out the people that need it the most? That is my biggest concern with this, folks, is that we, like Colorado and like Oregon, make this so complicated that the people doing it have to charge so much that we price out the people that need it the most. Again, you know, the ancestral users of this are where we should get most of our information on dosing from. I believe that wholeheartedly. Secondly, I'm still seeing a lot of comments about microdosing. And I agree 100 to 150% that we should be allowing microdosing. However, just like in Colorado that Adrian talked about earlier, and I had this conversation with Dominic before we even broke out into subcommittees, the way the bill is written, and we can only stay within the guidelines of the way the bill is written, just like Colorado though, if they're going to do any dosing, it has to be done at a clinic and they have to stay for a certain amount of time, which unfortunately makes microdosing near impossible because nobody's going to go in and stay in a facility for 2 hours every day to do their microdosing. Can we possibly in the future get a new bill or a change to the bill to where we can allow it? Yes, but let's work on that after we get this program up and running, rather than, because, and Dominic, please step in if I'm wrong. If we want microdosing now with this bill, would we not have to send this bill back to have them rewrite it and send it back to us?
Yeah, just to answer that, Shane, is so with the law, the way it currently is the dosing does have to happen at approved locations. Now, we are trying to figure out some ways that that can be done with regard to hospice and similar situations, because obviously you cannot have somebody who is confined to a bed or otherwise is not able to be mobile to, ask them to go to a clinic even down the street from them. And so we are trying to figure some of that on out, but it does make it very impractical with regard to microdosing because of the issues that they have seen in both Oregon and Colorado. That doesn't mean that it can't necessarily change down the road, but for the very beginning, and Eric, I appreciate that about the doctors for to approve the home. There's a lot more than just saying we approve the home. There are safety issues that I know a lot of people would say or may say, well, why do we have to be worried about that? But there are. All it would take, for example, is one caregiver running into an issue, getting hurt, that could potentially create issues. So we have to be a little cautious with that because we want to ensure the longevity of the program. So we want to do things the right way so that it doesn't end up having a problem to get shut down quickly and then us not to be able to help anyone. So we need to make sure that we're doing things in a way that fit within the structure of the bill and also within safety so that we can ensure there are not problems that create it. Do I think there are ways to do that? Yes. Will that probably be a part of our initial launch in December? Probably not, but that doesn't mean we don't want those conversations so that we can figure out what those challenges, potential obstacles are, and figure out ways around them. But to get things started, we need to focus on, how is this going to happen within a clinic? Let's get that part moving forward, and then we can look at some of those other aspects, especially as we see how the hospice actually rolls out, works, and so that if we have to pivot on anything, we can do so quickly without it being something where we're taking things away from people. Because that's the last thing we want to do is start saying you can do this, and then we have to pull it back because of potential issues or concerns. So we have to make sure we look at that all clearly and well so that it doesn't become an issue or a problem. So the conversation is good. We want to overcome those challenges. We have to figure out how to do that. Our first step, though, is implementing the program in a way that we can. I hope that makes sense.
Yeah, and like I said, I just want to make sure everybody understands that I don't think anybody is disagreeing that we should be doing microdosing. It's just that we're not going to be able to do it under the current way that the bill is written in this first go at this. Yes, once we're up and running, or as we're getting this up and running, we can include facilities to include hospice care. It's very doubtful that we're going to get any approval on take-home microdosing, because no, the way it's written, I don't think that the approving body will ever approve that we're calling people's homes a approved facility. But we do need to work on that, and maybe in the next phase of this. I 100% agree, but I just don't want to see a lot of calories being spent continually talking about this when it's just not feasible in this first round of things.
Thank you, Shane. That's all of our hands. I'll just catch up on the chat. Eileen says, I do agree with giving practitioners a jumping off point based on what we do know. They can titrate up or down as needed, just like someone would do with any other medication to treat the patient. Like personally, I'm a lightweight, 0.5 grams of dried psilocybin cubensis is perceptual dose for me, and I'm not 90 pounds soaking wet. We know that it's not based on weight in any cases. I do love that there is great sensitivity for not creating rules that will later need to be changed as we learn more, which is often more difficult. We also don't know if there are other compounds in psilocybin containing mushrooms that have therapeutic benefit because there hasn't been a ton of research on it. We might learn that in the future. James added, sorry, typo, including them for the entourage effect, if most effective. And Eileen added, also, I know that the group that is discussing practitioner training is considering whether to allow those who have already been trained in programs designed for access in other states. When we talk about what to use for measurements, we need to know how others have already been trained if they are to be included. If what New Mexico outlines is different, it might mean that additional training on dose is required. And those are all the comments. And I don't see any hands up at the moment.
Just one item, Jorge, and for this, and Eileen, that's a great comment. I'm glad that you brought it up. We actually did already present briefly about the fact that if there is reciprocity, because that's still a question, although I believe that that will end up happening with regard to the training, that there will be a module that the department will actually create that will have that information regarding what New Mexico statutes and procedures, guidelines, how they differ from the other states. And so that would be a requirement that they would still be able to do, or it's that they would need to do to engage in that reciprocity. So that part has already been done. But exactly what that includes, we don't know yet, and that'll be a part for that committee. So thank you.
Alrighty, with no discussion, I think there is a few things that we were in agreement on. So if we are good with MTP and APF, we might take a vote to just approve those two. Then what I would recommend after is, I'm gonna say if you have a dosage levels schematic or framework that you would like to submit, you can submit that to the email that Dominic provided, that medical.psilocybin@doh.nm.gov. I will review them and compile what the most common are, as well as if you can provide scientific evidence, that would be great. But do we have agreement on MTP and APF? If you agree with adopting MTP and APF, can you please raise your hand? And
Currently, there are 9 hands up. And then,
All right, so with 9 hands up, then we have the adoption of MTP and APF. Those frameworks will allow us to still create those dosage levels. I admitted the first time that the dosage levels that I provided were not final. But I did want to at least have something that way when you guys are presenting your dosage levels and sending them to that email, we can have something to base off of, some unifying factor to create the system as a whole. I do think that the dosage levels need a little more work. So I am totally open for comment.
And
Chris.
Wait, before we jump to Chris, just to let everybody know, we had to lower everybody's hand because we couldn't tell if it was vote or if you had comment. So if you did have your hand up and it was for comment, please go on ahead and put it back on up. If it was just because the vote and it was still remnant hand being raised, don't worry about that. But anyway, all right, Chris.
2 things, like we just voted for, we didn't have a vote for those who were against, and can we vote on those 2 things separately? Because I think there may be some people that would like the MTP, but not the APF.
Okay. So let's just go ahead and redo the vote one more time. Please lower your hands now. If you would like to vote in the affirmative of adopting MTP and APF jointly, please raise your hand.
Okay, we have 10 hands raised at this time. Any
All
additional?
right, please lower your hands. All right. Are all hands lowered? Not yet.
No, we're working on that. Give us just a
Perfect.
moment.
Thank you. All right.
Okay,
If you
all
would like
hands
to
are
vote
lowered.
against the MTP and APF jointly, please raise your hand.
we have 1 hand.
With 10 in favor and 1 in objection, the motion is passed. MTP and APF definitions are adopted. For this, now we can go ahead and do the work of setting up the dosage levels. I do want to give time for the public to come up with those things. So I will, if there is no other comment, I will close the meeting, but I did see a hand come up at the last second. Gregory?
Greg, your mic has been enabled.
We can deal with this in another meeting, but I really would have liked, I asked to comment before the vote. I feel like I should have been given an opportunity to comment before the vote, and I feel very strongly about this issue. I think that I'm not against what we're measuring. I'm trying to influence the acronym. There is existing usage of PCN to refer to the exact same thing with the exact same calculations that you have set. I feel that PCN is a better term, and then for psilocybin equivalency, PBCE would be the correct term. That I just gotta say that I think that adopting the language of MTP is not the right move.
Okay. Well, I appreciate your comment on semantics, but if there are no other discussion, is everyone good to adjourn?
The only thing, Ian, the next meeting?
Oh, yes. I'm so sorry, guys. Is everyone okay with just keeping the schedule another 2 weeks? Would be, actually, hold on. Next 2 weeks would be the 13th. February 13th work for everyone? Raise your hand if you're good with the 13th.
Dominic, were we trying to hold off on further meetings until after session concludes?
Yeah, yes, and that's what, thank you, Kathy. I was just checking on the calendar. We are asking right now, Ian, for the committees to just hold off until that point. Session ends on the 19th. We do already have end of life care on the 20th, but there are, and the board meets on the 27th on those Fridays. But either on the, oh, I'm sorry, my calendar just decided to skip into April. But on the 20th, there is 3 to 5 is open at the moment, February 20th. And then on the 27th, the afternoon is completely open as far as committee meetings.
Alrighty, so would you guys be okay with 1 p.m. on the 27th? Raise your hand if you are good with that.
And there's 6
6
hands up at the
hands,
moment, 7.
7 hands.
Okay.
6 hands. And then, or 7. Six. Okay.
Well, some people had lowered their hands, so I think we ended up with about 8 or 9 total.
Yes.
Okay. So then we will do the 27th at 1. I look forward to seeing you all there.
And please submit any public comments by email by February 2nd, please.
And also your dosage level recommendations. Thank you, guys.
Thank you.
Oh, can I also add for submitting comments for this committee, if you could just indicate specifically that it's for this committee, the dosage administration and clinical practice.
Yes. Alrighty. Thank you, guys. Have a great rest of your day.
Thank you, everybody.